Hi guys, so this is the first of the recordings that I'm going to do for you for the Chapter 19 PowerPoint based on the questions that I've seen in Blackboard in the discussion board so far. I had a few students ask about the blood breakdown and the composition of blood, and I wanted to walk through that with you. I'm going to turn off the video of me just for a second so that you can actually clearly see all of the things on this figure. And I'm going to walk through that for you for just a second. So the first thing that you need to pay attention to here is the, get black here, is the test tube. So if you imagine that someone has drawn some blood, they've put it in a test tube, stuck it into a centrifuge. Once that blood has gone through that centrifuge, what happens is that the different elements that make up whole blood will then separate out based on how heavy they are. So everything that's at the bottom is going to be the heaviest, everything at the top is going to be the lightest, and then in the middle you have sort of the middleweight guys. 45% of what you have in whole blood is going to be the formed elements. Now, somebody asked on the discussion board what the formed elements were, and they are the three types of blood cells that you talked about in your lab, the erythrocytes, the leukocytes, and the thrombocytes, or the platelets. So here you have the erythrocytes, here you have the leukocytes, and here you have the thrombocytes. That would make up the formed elements in the blood. They're cell-like, but they're not true cells. That's why we don't refer to them as that. We call them formed elements instead. Within the formed elements, in the white blood cells, you have multiple kinds of white blood cells. And you do need to know the names of these. You do not need to know the percentages, but you do need to know the order that they come in terms of most to least or least to most. So if I were to ask you which of the white blood cells has the highest percent or takes up the highest percent of whole blood, you would want to answer neutrophils to that. As opposed to least, you would say basophils. Platelets, you need to know exist within the whole blood, but you do not need to know the ranges for any of these. Now, the platelets and the white blood cells are in a separate layer that we refer to as the buffy coat. It is in between the red blood cells and the plasma. The plasma is mostly water. 91% of the plasma is water. And the rest of that is proteins and albumin, or proteins and other solutes. Now, if we move forward to the next slide, we have the same figure. You see that I've broken down in the text for you the proteins and explained to you what each of those proteins are, as well as given you an explanation for all of the other solutes that make up 2% of the blood. So this would be 2%, this group up here would be 7%, and the rest of what you have in the blood would be water. You need to know the names of the things that you would find in each of these areas, and you need to understand the descriptions of them in the text. You do not need to know the percentages of each of them. Somebody also asked a question on Blackboard about what a colloid is, and plasma is a colloid, and what that means is that the liquid portion of blood is thicker. Think about, when you have a cut, how thick blood is and how sticky it can be. And that thickness and that stickiness is what allows the proteins and the other solutes and the platelets and the white blood cells and the red blood cells to stay suspended within that solution and not fall out of solution as they're moving through the blood. That is going to be the end of this particular video in relationship to the plasma. If there are more questions about this concept after you've watched this video and reviewed the information on the PowerPoint, feel free to add another question in the discussion board and I will see if I can help you out.